Here's another technique, another methodology which has also fallen out of favor. But I think it's a good time, for you to at least understand what's available. And a large amount of bioinformatics effort is being put into the processing, the analysis of next generation sequencing data. A lot of bioinformaticists spend a lot of effort into GWAS, Genome-Wide Association Studies, okay, also known as Whole Genome Association Studies, WGAS, also known as Common Variant Association Studies. So, what were these studies about? Again, this is all genomic level work. Genomes vary between individuals in different ways. The differences are in the DNA sequences. These differences can be a single nucleotide polymorphism, and if you remember the video from the use of the genome browser, whether it's the Ensemble genome browser or the Santa Cruz genome browser, you'll realize that there is a resource which actually identifies single nucleotide polymorphisms. So GWAS genomes vary between individuals in different ways. Genomes vary between individuals in different ways. The differences are in DNA sequences. These differences can be single nucleotide polymorphism. A single nucleotide is different, but this can result in a different protein. Remember, in the coding region, in the open reading frame, if you change one nucleotide, you have a different codon. If you have a different codon, you have a different amino acid, which causes a variation in the protein. And depending upon the structure, functional aspects of that protein, if that particular amino acid is pivotal, it can change function. Okay, deletion. So if you have a missing nucleotide, that results in a single nucleotide to a large section of DNA that is missing from a chromosome. Genetic disease results from deletions. Frame shifts also result if you have a deletion. Remember frame shifts, right? When we talked about open reading frames, when we looked at, if you remember the video on the use of the translation resource at XSpaCy, you might have an insertion results from one or more nucleotide base pairs that get added to a chromosome. And then you have copy number variation. Some genes and chromosomes have more than the requisite number. These might result from insertions or deletions. And I wanted to mention one study that I worked on. I did a little bit in terms of bioinformatics, wrote 2 or 3 small pieces. This was a study on identical twins, monozygotic twins. And you typically say, well, monozygotic twins that are identical in every respect, should really have identical genomes, and they don't. And it's not just the SNPs that are varying, but also the copy number of the genes that are varying. And this was a pretty interesting study. It got mentioned in the New York Times, and I was glad to be part of that study. Okay, so this idea is the idea that a single nucleotide polymorphism, for example, can result in a disease was obviously simplistic, because now we begin to understand that that you have SNPs that are in various parts of the genome, and these are then correlated, and a disease as a result of several SNPs and several other things coming together. So the idea that one disease resulted from one SNP was simple. If you look at the National Library of Medicine, National Institute of Health resource, and just to look at the definition of GWAS, genome-wide association studies are relatively new, so obviously this is a little outdated. It's a way of scientists to identify genes involved in disease. Searches the genome for small variations called single nucleotide polymorphisms, SNPs, that occur more frequently in people with a particular disease than in people without the disease. Each study can look at hundreds of thousands of SNPs at the same time. Researchers use data from this type of study to pinpoint genes that may contribute to a person's risk of developing a certain disease. Okay, also fallen out of favor. A GWAS is phenotype centric. That is, you identify based on an expressed characteristic. So, 2 groups of individuals are selected. One possesses a specific trait and one which does not. The one which does not is called the control group. This trait could be, a disease. The common applications for this GWAS have been clinical. So the DNA of the individual is collected, purified, etc. Then you create a probe set of these SNPs, and they're called SNP arrays. There is a database in GenBank or associated with the NCBI called as a SNP array, DB-SNP. This database is called DB-SNP. SNP arrays are composed of probes approximately. So once again, the SNP array now uses microarray technology. SNP arrays are composed of probes approximately 25 nucleotides long. There are pairs of probes corresponding to each allele for a SNP. So what is an allele? An allele is a variant form of a gene. Some genes have a variety of different forms, which are located at the same position or a genetic locus on a chromosome. Humans are called diploid organisms because for each genetic locus associated with each trait, we have two alleles. Each allele comes from one parent. Each parent of allele represents a genotype of a specific gene. Genotypes are described as homozygous if there are two identical alleles at a particular locus, and heterozygous if the two alleles differ. Alleles contribute to an organism's phenotype, which is the outward appearance of the organism, and not necessarily outward. You might have, for example, a proclivity towards suffering from a particular disease is also a phenotype. So the remaining steps of DNA hybridization, you create a SNP array where the probes are pieces of genes associated with SNPs, okay, which have a SNP in them. And everything else is basically experimentally like that of a microarrays. The measure of relative intensities for a test, a control versus a diseased individual, for example, are sent through statistical processes. Some statistics associated with microarrays and therefore GWAS, allele frequencies, the frequency of the occurrence of an allele for the population being studied. It is a measure of variation at a locus in a chromosome. Odds ratio are calculated. The odds of an individual with an allele having a disease against the odds of an individual who has the disease without having that allele. This odds ratio, and now you begin to understand, right? We've talked about odds ratio in terms of, having something and having the disease, having it, not having the disease, not having it and having the disease, and not having it and not having the disease, which is the rest of the population. You understand how to calculate the odds ratio. This odds ratio can be used to determine the association of a gene variation with a disease. And once again, I'm not going to do this because we've studied this ad nauseum, but I think this slide is a good time. It's good to recognize the slide. And those of you, eventually what happens, the multiplication is, you multiply cross, and that's divided by, and that's how you determine the ratio. Just in case you don't remember at the last minute. 30 times 50 and 50 times 70. Okay, that gives you 0.42 of possessing an allele versus. And I'm not going to talk a lot about this, but I think again this is something that also was very popular. A haplotype is a group of genes within an organism that was inherited together from a single parent. So the word haplotype is derived from the word haploid, which describes cells with only one set of chromosomes, from the word genotype, which refers to the genetic makeup of an organism, right? So haplotype is basically the entire kind of a set of genes that came from one parent. The 1000 Genome Project presents a map of the normal human genetic variation, everything from tiny changes in the genetic code to major alterations in our chromosomes. Okay, this is from the 1000 Genome Project announcement. In a DNA version of Spot the Difference, Emble scientists and their colleagues studied genomes of 1,092 healthy people from Europe, the Americas, East Asia, systematically tracking what makes us different from each other. And that is an integrated map of genetic variation from 1,092 human genomes. So by characterizing the geographic and functional spectrum of human genetic variation, the 1000 Genomes Project aims to build a resource to help to understand the genetic contribution to disease. Here we describe the genomes of 1,092 individuals from 14 populations, constructed using a combination of low-coverage whole genome and exome sequencing, By developing methods to integrate information across several algorithms and diverse data sources, we provide a valued haplotype